Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Dr. Andrea Gambotto. He's an associate professor of surgery, University of Pittsburgh School of Medicine. And we're going to talk about SARS-CoV-2, aka the coronavirus, the Wuhan virus, and a potential vaccine. So, Andrea, thank you for coming.
Thank you for having me in your podcast.
Yeah. So tell me what specifically you're working on in regards to a vaccine for SARS-CoV-2.
We're working on potential vaccine candidates for COVID-2. We have been doing this now for many years. We are trying to develop a vaccine for emerging infections. And in particular for SARS, we were the first group to develop a vaccine in 2003. It was the first coronavirus vaccine ever developed for humans. That vaccine was never tested because the first SARS disappeared early in 2004. And of course, there were no funding, no resource to move our vaccine into clinical testing. Then we work on other emerging infections, and in 2014 there was another coronavirus, the MERS coronavirus, Middle East Respiratory Syndrome coronavirus, and again we developed a vaccine back then. Did some preliminary testing in the animal models, and then tried to move that vaccine into humans, but again, there was little or no interest to do that. And again, now we are in a few years later, we have SARS coronavirus 2. And again, we develop our vaccine, a prototype vaccine. We actually developed more than one prototype, and we are we have tested in animal models, and we try to move this vaccine into the clinic now. Hopefully this time will be the time that we can test our vaccine into people. We hope to do this in the short period of time because we are manufacturing now the vaccine, applying for the clinical study through the FDA.
Well, hopefully this time they won't suddenly lose interest, not because of the coronavirus going away, which would be nice, but hopefully they won't lose interest in general, so you're not stumped yet again.
Yeah, I think this time is real. It's the real time. So now is the time to test those vaccines, and our is not the only vaccine out there. There are many other vaccine candidates. But again, we have some experience with this bug now with the coronavirus. I think we should have a chance to try our vaccine, which we think has some future that may be convenient and may be a good vaccine candidate.
What's unique about your vaccine? How does it work?
So the unique is that, first of all, we developed several platforms. So for many years, we work on adenovirus-based vaccine, and that's now is one of the most hot platform for this particular coronavirus. And we do have a vaccine candidate based on the adenovirus platform. But the one that our pipeline, the first one that we're going to be testing is a subunit protein. And the difference between the viral platform and the protein platform is that the protein platform is it's safer, it's perceived safer, and it's safer because it's like the influenza vaccine. Once you manufacture, you figure out the way you manufacture, streamline and scale up the production. It's easier than other platform. So it should be easier to manufacture in large scale. It should be easier to administer. And the proteins-based vaccine, in particular, the way we deliver the vaccine could be potential stable at room temperature of low, relatively affordable temperature like 4 degrees. And this is very important for if you think the vaccine that needs to be distributed and used all over the world in place where the refrigeration could be a potential problem, or just the delivery, you may interrupt the cold chain where most of this vaccine need to be stored. So these are not some of the consideration. And so the way we try to deliver, one of the way we are trying to deliver this subunit protein vaccine is with the patches. These are microneedle patches that we formulate are based on sugars. So we dissolve the vaccine in sugar, and we mold the vaccine into these microneedle patches. They can be self-applied, and they dissolve. They basically have they go in the superficial layer of the skin, so they don't penetrate a lot. It's not like a needle injection. It's just it goes in a part of the skin where there is no vessel, so there is no blood, there is no nerve, so there is no pain, so there is no blood, no pain, and you can self-administer. So if it works, a platform like this is ideal, not only for this vaccine, but for every vaccine, because it would be very easy to implement all over the world, a vaccination with this platform.
Yeah, I spoke to another contributor that talked about the microneedle array and how it dissolves, and that's great. Yeah. The protein, what is it? Part of the capsid? That's or is it the spike protein? Like what kind of proteins are going to be put into the microneedle array?
Well, we have the spike protein, which is the protein that is used by the virus to get into the cell. So we use this spike protein, and the spike induce antibody, and this antibody cross-react, of course, with the real virus. And once they bind the real virus on this protein, the virus is blocked for entering into the cell. So they are called like neutralizing antibody, because neutralize the ability of the virus to enter into the cell.
So you think this will be neutralizing, not just binding?
Well, it's no. The most of antibody against the spike protein are neutralizing antibody. There are some antibody that won't be able to neutralize, but let's put the most of antibody that are generated against the spike protein should be able to neutralize the virus entry. Some may neutralize more efficiently than other, and that's still things that people are trying to, ourselves as well, try to understand in what extent the binding antibody neutralizing. There are some data that virtually all the people that develop a spike antibody, those spike antibody have a neutralizing capability.
Yeah, what happens when you have just binding versus neutralizing? What do you think differentiates the mechanism?
Well, it's dependent of the so this protein is used by the virus to enter. And there is a spot of this protein that is the part that binds the receptor in human cell, which is called AC2 receptor. So if you have antibody on the binding interface, of course, that one are going to be more effective as a neutralizing antibody. But there are some other antibodies not binding that interface, but can still inhibit the entry, the binding of the virus to the receptor. So it doesn't need to be on the hotspot. And I would say 80% in the way I see, envision the binding and the structure of the spike protein. I would say 70-80% of binding antibody are going to be able to neutralize the virus. And since when you give an antigen, you're not going to develop only one antibody. You probably develop 300, 400 different type of antibody. 200 of them are going to be neutralizing. And you have a few, 50 they want, but it's okay. There is other mechanism for which antibody can have some effect, immunological effect. So every antibody counts.
Okay, I didn't realize that. Do people profile the antibodies that are produced, and out of the population, what percentage are neutralizing, what percentage are binding? Do you get a distribution? Have you observed that with different vaccines?
You cannot really do that, because every time you immunize an animal or a person, you're going to develop different type of, so you develop a pool of antibody. And if me and you get the same vaccine, we're going to develop, you're going to develop your 300 antibody. I'm going to develop my 300 antibody. And they are unlikely to be overlapping, but I would say that 200 of each of us are going to be a neutralizing antibodies. So the thing is that this antigen should induce in 100% of the individual neutralizing antibody. Now, the potency of this antibody, so how much neutralization you have depends on the level of antibody. So more antibody, more neutralization, and then depends how much virus you get infected with. So these are the type of things that you don't know until you test the vaccine into clinical study that determine what's the dose of your vaccine. And first of all, if it's effective, and then the dose of your vaccine, because more vaccine, more antibody. And then some of these antibody have different longevity. So some antibody lasts for 2 months, some antibody lasts for 1 year. So of course you want to try to develop antibody that lasts for 1, 2, 3 years. Longer, is better. And but this type of information cannot really be determined even in animal model, because they are not very informative, the animal model. When it comes to antibody specificity, because every species is going to develop prevalently a type of a group of antibodies. But you can generalize saying that since the response is polyclonal, so there are many types of antibody, the most of them should be able to neutralize the virus. But this is the hypothesis, and it needs to be tested then in the real. And there is no way really to test unless you spend the next 50 years to dissecting the immune response. So nowadays seem easier, but it's not worth to spend the years and years to try to dissecting the response, when you can easily do it in a phase 1/2 clinical study. And that's what we.
I had thought that sometimes antibodies they're able to allow the virus to change so that they actually assist where the virus gains entry through a different mechanism that's more effective and more deadly. I believe with, dengue fever or there's a couple of conditions.
That is called an enhancement effect, but it doesn't.
What's it called?
Enhancement effect of an antibody. There are some disease, there is historically a vaccine, the respiratory syncytial respiratory virus vaccine that was developed in the 70s. That was indeed inducing this antibody, which were binding, but were favoring the entry. And dengue is another virus that does the same when it changes serotype, when it crosses serotypes. So, but for the SARS, there were just one study, couple of studies that shows some possible enhancement, but that was demonstrated in vitro, never in vivo, and it was never clear, considering that there had been hundreds and hundreds of vaccine studies of coronaviruses looking for enhancement, and there are only a couple of studies that were basically not reproduced by other groups, and then if you see so if antibody that favor the entry of the virus would have been the case in the spike in immunity, that would also be happening naturally. So people get infected, and then the first infection, that's what happened in dengue. You get the first infection, the second infection is much worse. And people die during second infection because they get infected with one serotype, and then the second infection with this different serotype trigger this enhancement effect. The million people infected with SARS we would have observed clinically this effect of people that recover from the infection, and then a second infection was much worse.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Do you think that's rare, this antibody enhanced?
Yeah, I work on many viruses that potentially can induce ADE, and I don't believe that this coronavirus is going to be one of those viruses that induce ADE, because we don't have a single evidence. People have done some, we have done some vaccine study where we haven't done challenge studies so far, but people have done challenge. They have used a spike protein, so they have not observed any ADE in this preclinical study. The published and the one that we know, the report, and also the vaccine that are in the earlier stage of testing in humans. Apparently, they are not inducing enhancement, and the reason is we don't know that is not the case. But when you see a vaccine moving in phase 3, you pretty sure that they have done some testing to rule out the enhancement. And if somebody else is using spike protein and it doesn't show enhancement, our vaccine or all other vaccines are not going to induce enhancement. That's basically the message here.
So what have you learned from working on coronavirus vaccines, through many different, for SARS and MERS, and what interesting uniquenesses are there to coronaviruses and vaccines today?
Well, we learn a lot. I learned a lot, and consider that when I started in 2003, there was this new bug that came out, and it was, the mortality was very high, and we have no idea what was the antigen. So now everybody knows that you have to use spike. But why we choose spike? Because back then we test all the antigen that the coronavirus added, all the structural protein, and we determined back then in 2003 that spike was the protein. Then we tried to use spike, just the S1 versus the all, and we learned many other things. So it's been a journey, and we learned a lot. First, we learned the most important things. We learned that spike is the protein to target. But now, we in some of our vaccine prototype, we are using a nuclear protein, which is also important. Nuclear protein because give you an immunity against it's not an antibody immunity, but it's a cellular immunity, which could potentially cross-protect with other future coronaviruses. For instance, the nucleoprotein of SARS-1 is almost 90% conserved to the nucleoprotein to the SARS-2 that came out, appeared 17 years later. So we have developed a nucleoprotein vaccine, based T-cellular vaccine for SARS-1, which now is likely going to work for this immunity. What a T-cell vaccine would do is not the person that get infected will still get infected, but the disease will be much milder. It's probably not going to kill anybody because you have T-cell that can take care of the virus. Is slower than antibody because antibody just block the infection. T-cell have to go and get the virus from inside the cell when the cell already being infected. But it's very effective also mechanism. So we are now, with this knowledge, we are combining this knowledge, and we have one of our vaccine contain both antigens, and we are advancing all this product, all this vaccine, of course, one at a time. But we have the version 1.0, 2.0, 3.0. So we are advancing also in the manufacturing those products in house and prospective.
Are you gonna? So you're doing both a novel vaccine, but also a novel method of administering it with the microneedles. Are you pushing ahead separately on both to make sure that this happens as soon as possible?
Yeah, we are using we are pushing two different trials. One is with an adenovirus, which is classic, which I have to say, I was the first one to develop an adenovirus-based vaccine, and since I like it and I know that it's potent adenovirus, there are several limitations of adenovirus. Is going to be one shot. So once it's for this coronavirus, we cannot use for other coronavirus in the future. The protein is something that we can build, and it can become the technology for future coronavirus infection. It can be used over and over and over again because it's like a flu. Every year you make a new one, whatever. Every year it's not going to happen every year for a coronavirus, but every once it's needed, you know how to do it and what to do it. So, and the protein can be done over and over. Adenovirus cannot be done over and over. So you are limited to one shot. And we do adenovirus now because we think this is an important shot now. So if you have one shot, this is probably the disease that you want to use your shot. So we're going to use the shot with adenovirus, with the protein. And if they both work, we're going to be happy. One works, the other doesn't. So we have used the best shot now. And then we work on the other one.
So what's your guess on how long to get through clinical trials for for your vaccine, the SARS-CoV-2 one?
Well, no, we are not a big pharma. We are just an academic laboratory. We do everything in-house. We discover the vaccine. We manufacture the clinical grade vaccine. Well, we do preclinical testing in the mice and other animals if are available. And then we have to manufacture the vaccine. We have to write the clinical study, organize clinical study, and then put this through FDA, get it approved. So our speed is not a big pharma speed, but I think we can come up with the products which are like more creative than big pharma. So we can take some chance that big pharma cannot. And if they fail and we succeed, we can be the plan B or C, or we can be the plan A if our vaccine show an effectiveness and an easy to manufacture, to administer. That when we do this product, we always think that these are going to become potentially a 1 billion dose product. So we have in mind that we can produce a vaccine that can produce it only in 100 doses, and that's it, because that's one of anybody. So this protein-based, adenovirus-based platform are platforms that are robust and can be scaled to the quantity. So we don't have the capability here, but that's not our goal now. Our goal is just to show that these are effective and they can be manufactured. And if that's the case, we're going to give this product to the big pharma, and they know how to manufacture. And so just we want to make sure that we develop compatible technologies with big pharma. And then they will be the one that will come and say, okay, I want to use your vaccine. Timing wise, we open 2 months, 1.5 months to be into the patient. We are manufacturing as we speak. So we are releasing our products. And as soon as we release and we get the green lights, we should be able to jumpstart the trial.
Why is it? Why does everyone say it takes years and years and years to make a vaccine? What are some of the hurdles to it? Why does it seem to take so long? Because
there are many things that are not that until you do it, you don't know what is going to be the end point. So when you make a prototype vaccine to be used in the lab for mouse experiment is something. When you try to implement this product to become a clinical product, to become a scalable product, that is when the problem comes. So what if you make your vaccine and the yield of your vaccine is 100-fold less than a normal vaccine on which you just base your technology? Then becomes a no product, because if you struggle to make 100 million doses for any vaccine now, 100 million doses, if you have 100 times yield reduction, you can make only 1 million dose of vaccine. So if that's the limitation, you don't want to produce, you don't want to even take your time to investigate a million dose vaccine, because it's never going to be the vaccine. So you have to make sure that you can manufacture. And this particular vaccine is kind of tricky because spike protein is not the easiest protein to produce. It's not the easiest virus to manufacture. And the hurdle, some of the hurdle that we encounter, but we encounter some of those hurdles also in the MERS and the first SARS. So we knew that. So, but some people may not be aware that making an adenovirus that encodes spike is not going to be the same as making an adenovirus that encodes the hemagglutinin on influenza, because the yield is going to be much lower. You have to deal with that, so you have to try to increase the expression of the spike protein, so you can afford a reduced amount of vaccine. You have to develop a way to deliver the vaccine that are more efficiently. So if you increase the efficiency of 5%, that make a big difference. So these are the type of development study that you have to do. Sometimes people or a company jumpstart on production and jump and say, okay, we are the first, but then you have to go back and you do your own homework to understand what went wrong. And probably some of the player in this field, and they pretty soon they're gonna go back to the lab. And we are trying to take a little bit more time and just make sure that when we are there, we have a solid product. And if we have, and if it works, and at the end, we have used our experience to our advantages. But if we could have used our vaccine two months ago, and we developed, we published, we had it ready, we did the first lot, but we say, okay, let's take care of these 2-3 things that we think are going to be important down the road. We don't want to go back and repeat our phase 1, phase 2, or we don't want to fail because we were sloppy to begin with. So there are many things that make the vaccine a complicated task to perform. It's not as easy as one can think. There are too many variables that needs to be taken into account. And somehow you need some experience to deal, to prevent things, to limit the implementation of a later stage for the vaccine.
So what governs yield? Can you talk about that at all? Like, why would you be able to, culture one tremendously, but not another? What are some of the factors that go into it?
So yield is everything. So if you do your own work and you can increase the yield of 100-fold of a product that is difficult to produce, then you are in a good situation. So yield here, we talk about 100,000 of liter of manufacturing capability that only big pharma has. But some of the vaccine capability now production is like 300 million doses. Let's say for an adenovirus, it's 300 million doses. Now the problem is that when you do a phase 1 study, phase 1/2, you use 2 or 3 doses. And these 2-3 doses, the lowest dose and the highest dose is 100 times apart. So then, of course, if you can afford to use the lowest dose, now we have 100 million doses. If you have to use the highest dose, then you're square one because you have only 1 million doses. And 1 million doses in 6 months, you don't have a product, you don't have the vaccine. And so these are the most important. The dose escalation that you do in this phase 1 study will tell you whether your candidate vaccine is a good candidate vaccine. At the same time, you have to work on different aspects of the vaccine. You have to work on the yield of production. So can you increase the amount? Can you make instead of 100, 1,000, whatever is that 1,100? Can you increase the expression of the transgene that you put in this vaccine 10 times more or 5 times more? Because that reduces the amount of vaccine. So if you combine 2 or 3 of these things, you can get 1,000-fold reduction of the need of the vaccine. And these are the type of homework that you do for these particular vaccines. So you have to make sure that you can make it, and because making a vaccine in quantity that are not, that don't satisfy a large population is not the goal for this particular vaccine.
And then the microneedle array, will that boost the efficacy of a given vaccine, or what's the data show so far?
Yeah, so that's the rationale for us to using the microneedle, because when we were studying MERS, so we test the microneedle array on MERS coronavirus. And what we found is that when we compared the just needle injection versus microneedle, the microneedle was much more potent, and you need probably 1/5 to 1/7 of the amount of vaccine when delivered with microneedle when compared with intramuscular administration of the vaccine. So that is one factor of reduction of 5-fold. You need 5 times less protein, and if you can afford if that will compare with influenza. Now, influenza with 2 microgram given with microneedle, you can immunize a human being. That means that with 2 kilogram of this protein packaging in microneedle, if microneedle is as effective as influenza, with 2 kilograms, you can immunize all over the world, all people in the world. So 2 kilogram is 4 pounds. 4 pounds is not that much. It's doable. And these are the type of number that we have in mind. So we have from 2 to 200, 400 pounds of vaccine. It's unlikely that I'm going to be able to produce in my lifetime, because you need a monster bioreactors that they are probably not available in the world, given the yield of production of the spike protein. So spike is not a protein that you can produce in like an antibody, like other protein. It's kind of the yield is limited, at least in our hands and other people's hands that we know. So everybody is having some difficulties in producing a large quantity of spike. But we improving day by day the production. We know that we can get to a certain level, and but again we have to, on the other side, we have to reduce the need of this protein, and this will clinical trial will tell us how much we need, and then we can understand whether we have a vaccine. So we have a platform that is doable. And again, you have to buy. So because if syringe delivery is not as different than microneedle, of course, syringe is preferable because it's an already implemented system. It's much easier to just put the drug into the syringe, that could be a better solution. At the same time, if you want to make package tomorrow 2 billion doses of vaccine, you're not going to be able to find anybody that can provide 2 billion vials to vial your vaccine, because there is no capability of producing anything in the billion quantity in the world. You're not going to find 2 billion syringe tomorrow. So that's the reality. So that's the advantage if you find an efficient way to do
the sweet spot. You need to, a million there's a sweet spot. 1 million may not be enough, but billions is too much.
1 billion is too much. It's too much for anything. So if you want to buy 2 billion can of a pop, you can't find it because the big brand don't produce that much. So it's right. No, billion is a big number. It's easy to say. Now you can say $1 billion because it's easy. We think right. That, but $1 billion of a single thing is a lot. So so you have to develop devices that are that you can manufacture, or you have to develop multiple devices. So one of our vaccine, the plan is to give it with the nasal spray. And if you can make a device that makes 100 spray, 100 puff, so you can deliver 100 vaccine with one device, and make a big difference. Because from 1 billion, you need now 10 million. 10 million is easy to handle. Well, it's not easy, but you can handle them. So these are the type of research inside the research. You don't not only need to develop a vaccine, you have to think how you're going to implement this vaccine, because you don't want to be sorry in 6 months, 8 months from now that you have a vaccine and say, oh, who's going to make it? How I'm going to deliver it? And then you're looking another 2-3 years to maybe realize that probably the vaccine was not the best vaccine, the best platform. And so this is kind of experience that guide, your choice, going one direction instead of the other. Sometimes seems to, one direction can be counterintuitive, but, and if you had a particular experience going in the wrong direction, so at least what not to do.
Okay. Well, very good. Andrea, what's the best way for people to find out more about the microneedle array and about the SARS-CoV-2 vaccine as well? They just go to University of Pittsburgh, or what's the best way to find out more?
Well, we're gonna probably publicize the trial whenever we will start, so it will become public. So, but we have a website at University of Pittsburgh that tell a little bit about the vaccine, and as soon as it will be starting phase 1, there will be the call for volunteer participate in the trial. Initially, it will be a limited amount of a volunteer, but then when we expand the study, we will need more and more people to join the trial.
Okay, well, very good. Well, Andrea, thank you for coming on the podcast. I appreciate it.
Thank you very much.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.